The BIOPER was one of the first studies, a prospective one, exploring the activity of palbociclib as second-line treatment following progression on palbociclib regimen. So the question was about if we can maintain the CDK4/6 inhibition as a major objective in not just for one only endocrine therapy treatment. And the important thing about this study is that it was based on biological sub-analysis. So the very clear concept is patients could go on the study if we have biopsy at the time of progression to the first-line CDK4/6 inhibitor, as well as other samples of blood sample, as well as tumor from the primary origin. The interesting finding is that about 1/3 of those patients that were initially sensitive to palbociclib then progressed and were retreated again with a palbociclib regimen, benefited again from the combination. So about 34% of patients achieve a clinical benefit to the second-line endocrine therapy and palbociclib. And that's good because it's in line with other options in second, third line following CDK4/6 inhibitors, as it has been shown for alpelisib and so on. What we have identified in the molecular sub-studies is that we can start to learn about very early mechanisms of resistance to the CDK4/6 inhibitors, and particularly high expressions of cyclin D1 as well as cyclin E were more related to tumors that didn't benefited from subsequent treatment with palbociclib, as well as those tumors who have a low ratio of retinoblastoma phosphorylated protein versus non-phosphorylated, indicating that these pathways could be related to a resistance to palbociclib.